if I could have your attention, it would be my pleasure to introduce Leah Guthrie for her visit this morning and the presentation that she's going to give us today. So Leah got her undergraduate degree at Swarthmore, a combined degree in biology and education, which I thought was really cool, and then went to Albert Einstein, where she was in a systems and computational biology program. And she began her work on the microbiome drivers of variation in terms of drug and dietary responses, and then continued that work in her postdoc at Stanford in the lab of Justin Sonnenberg. And I could take a big chunk of her talk talking about her accomplishments and awards, but I won't. She's an HHMI Hannah Gray Fellow, and the other part of her CV, I think, even more than the awards, was that really impressed me in particular were all of the leadership opportunities and the outreach activities and professional development activities. So Leah helps run grant writing workshops for colleagues at Stanford, and is a member of is an instructor in the software carpentry program, and many other activities. And so is really contributing to the field of microbiology as a whole. And we're all excited to hear your talk. So thank you.
Thank you for that introduction. Okay, there are chats. Okay, well, yeah, so thank you for introducing me. Hopefully everyone can hear me okay. Yes. And so I am really excited to have this opportunity to share and discuss my research with this group. And I am a microbiologist and systems biologist by training. And broadly, I'm really interested in understanding how human gut microbial metabolites shape our biology. And so for today, let's see. So for today, I'm going to start off with just a brief introduction that really gets at the question of why decoding kind of how diet shapes microbiota dynamics and metabolism, and in turn produces these metabolites that alter host cellular responses is actually a really important question and area of study, but it also is a really, has some really big challenges that I think require some new tools, and I'm going to talk about what those tools look like to me, and I think that also requires some specific priorities. And so I'm going to get into that. And then I'm going to talk about kind of one of the ways that I see myself addressing this challenge first. And so first I'm going to talk about my role in really getting at the questions of, well, how does diet shape variability in microbiota dynamics and metabolites? And then I'm going to talk a bit more briefly about another aspect of my postdoc work, which is really focused on understanding how these metabolites alter host responses. And then in the end, I will wrap up by really just focusing on what I see myself doing in the future and kind of what I think is most exciting about the types of questions that I'm asking. Okay, and so to just start us.
Okay, can you just click that little message up at the top so that your titles aren't obscured?
Yes. Let's.
Whack-a-mole here. That would be a high-risk maneuver, I think.
No. There's an X. Okay, I see it. Thank you. Perfect. All right.
That was the first.
Thank you. And so, okay. And so to get at this question of kind of, well, why is kind of diet and the gut microbiome so important when it comes to thinking about host cellular biology, I think it's really useful to kind of start off with this close-up of the gastrointestinal tract. And the communities of gut bacteria that live here and colonize the gut have collectively just this tremendous enzymatic capacity to produce numerous small molecules. And so, for example, our gut microbes can produce really complex metabolites de novo from primary metabolites. They can also modulate host molecules. But one of the areas that I really care about and what's really interesting to me is, and is also kind of a source of a large pool of the chemical diversity of gut bacterial metabolites, actually comes from bacterial metabolism of food compounds into a variety of small molecules. And as I go throughout this talk, I'm going to refer to these small molecules as microbiome-dependent metabolites, or MDMs for short. And so because a lot of these MDMs actually do not stay confined to the gut and instead are absorbed and enter systemic circulation, these MDMs have the capacity to alter various aspects of our biology. And so just as an example, a number of these MDMs are actually cleared from the body via the kidneys, and so they're filtered in the kidneys and excreted as urine. But when the kidneys fail, as they do as in the case of chronic kidney disease, a number of these MDMs accumulate in our systemic circulation as uremic solutes. And so here I'm highlighting a few of the structures of some of these uremic solutes that are also microbial in origin. And all of the ones that I've chosen to highlight are uremic solutes that are either associated or mechanistically linked to the progression of cardiovascular disease or chronic kidney disease in these chronic kidney disease patients. And what's notable about these MDMs is that their associations relate to kind of the fibrosis or scarring of both kidney and cardiac tissues. And so what I'm hoping that everyone takes away from these examples here is that a number of MDMs possess these disease connections. However, one thing that's really challenging in this field is that while you have some of these metabolites that possess these disease connections, kind of understanding this whole map of how you go from diet to some of these diet-derived microbial metabolites to altering host responses is quite difficult. And so I'll use phenylacetylglutamine as an example. And so this is a metabolite where we actually do know how it's processed. And so this is a product of kind of bacterial metabolism of diet-derived phenylalanine followed by host metabolism and modifications of diet-derived phenylalanine. And so we have really good insights into, we know what the metabolite is and we know its metabolism. And then on terms of host response, this is a metabolite where at higher levels, its phenylacetylglutamine can mediate a number of host cellular processes that are really important for platelet aggregation and thrombosis, and this can lead to increased risk of heart attack and stroke in these individuals with chronic kidney disease that also end up with these cardiovascular issues. And this metabolite carries out through this function by modulating host G protein coupled receptors, a number of adrenergic receptors. And so it's great that we know some information about how it affects the host, but what's kind of less clear about this metabolite is just the relationship between diet and how much of this metabolite we get. And so what's one thing that you notice in these patients with chronic kidney disease is that even among these patients, the levels of this metabolite varies widely from person to person. And that variability is really important because the more of this metabolite you have, the greater your risk of having these adverse effects. And so this is one example where it's really important, it would be really helpful for us to understand questions like, well, what is the role of diet in shaping the variability in these levels, both directly in terms of the amount of precursors available, but also indirectly in terms of how diet shapes microbiota dynamics that can in turn shape the levels of these metabolites. And so one thing that I hope you take away from the type of work that I do is that for me, the whole premise of my work is that really understanding kind of contributors to variability in microbial metabolites and understanding its impact on the host. So both function and sources of variability are the two things that I think are really important to interrogate about the microbiome. Okay, so with that said, I'm going to briefly get into the first part of the types of work that I've been doing, which is really thinking of this question of how does diet shape microbiota dynamics and metabolites. Next. And so when I first joined the Sonnenberg lab, like I said, as a graduate student, I've been working mostly on drug metabolism and understanding the role of the microbiome in drug metabolism. And I kind of think of things at more of a chemical-centric view. So I'm just like, drugs are one type of compound and foods are a different type of compound. And so like I kind of expected understanding variability to be, I guess, easier to sift through. But one thing that was really interesting when you follow the literature about the microbiome in diet is that while we have this general appreciation that diet shapes microbiome structure in terms of its composition, and that what you eat relates to the metabolites that you produce in your health, across studies, how much your diet contributes to variation in your microbiome, both community structure and other things, varies quite a bit. And so this was really interesting to me because our lab, as well as in a number of groups, do a lot of these dietary intervention studies to try to understand, well, what can you eat for a healthy microbiome? And the there was just so much variability. And so in trying to think about this question of, well, how can I come into this area and really think about how much diet affects the variability? And so I kind of think of things in terms of this triangle with these three kind of points. And so in terms of the factors that I consider as the key variables, you have diet, shaping, kind of variability in microbiome metabolites. And so imagine that each of these squares represents a different metabolite. There are some microbiota intrinsic factors that likely shape the variability in metabolites, and then of course a number of host factors. And so in order to really get at this question, we designed a dietary intervention study where we said, okay, we're going to keep diet the same, and then ask, well, when diet, the chemical makeup of what a whole bunch of people are eating is exactly the same, is that enough to control variability in some of these metabolites, or are we going to see an emphasis on some of these microbiota-dependent factors or other factors? And so to do this, we designed this dietary intervention study called the Microbiome Individuality and Stability Over Time study, or MISO study for short. And I just want to go over a few aspects of the study design that I think are really important to know. And so we started this study off with a baseline diet phase. And so during this phase, everyone ate their habitual diets, whatever they want. And we recorded what individuals ate through these really detailed dietary food logs. And then we switched to the intervention. And so this was a homogeneous diet. And so what this looked like for the participants is that for 7 days, for every meal, they ate what you see here. And I'd like to really note that with this diet is that everything here is in this like chopped salad format, and so it's all mixed together with the exception of the orange juice and the cookie. And the idea there was that to really make sure that everyone was mostly eating the real things and the same things. And so when we and others do these controlled feeding studies, yes, it's like you may keep everyone in a certain location and things like that, but what you'll find is that some participants will, you give them this meal and maybe they'll only eat the rice and they ignore everything else. And this was our way for really trying to homogenize the chemical makeup of what people see in their guts. And so we have lots of appreciation for our study participants that basically ate little packets of this for every meal for 7 days in a row. Okay, and so we had that homogeneous phase of the study for 7 days, and then we wrapped it up with a washout period where all participants got to go back to eating their habitual diets. And throughout this study, we collected detailed dietary food logs during the baseline phase and just kept track of how many of these packets individuals were eating. And then we carried out extensive metabolomics profiling. And so I have training in mass spectrometry. We have a lot of interest in mass spectrometry in the lab. And together as a group, we've really done a lot of work to try to quantify as many small molecules that are microbiome dependent as possible. And so this was a really fun opportunity for us to apply that pipeline to one of our dietary intervention studies. And then in addition to metabolomics that we did in participants' plasma, fecal, and urine, we also did some 16S RNA sequencing and metagenomic sequencing. And lastly, because of my interest in really understanding, well, what are the sources of variability in some of these uremic metabolites that I mentioned a bit at the top, I also did some targeted metabolomics just focusing on these 4 uremic solutes. And so one of the first questions that we asked once we got this study done, and I should say that this was a part of a larger clinical trial, is one of our first questions was, okay, well, for these known uremic solutes that are of interest to us and of our interest to our nephrology collaborators, to what extent does just controlling diet control variation that we measured in terms of the coefficient of variation across some of these known uremic solutes that we know a lot about? And so here, what I'm showing you is the coefficient of variation for these 4 metabolites of interest. And we were really interested in kind of the change in CV between the first study time point, which is in this baseline component of the study, versus the 4th time point, which is the end of the homogeneous diet. And what was striking to us is that for kind of the 3 known metabolites that have actual strong associations with the progression of chronic kidney disease, being on a homogeneous diet at least did not decrease their CV between time points 1 and 4, whereas for hippuric acid, which has some ties to renal fibrosis, but the evidence is less less clear is generally associated with health. For this metabolite, the CV went down significantly. And so we were really interested in that. And we basically did this analysis for all of the metabolites that we profiled. But I want to tell you a little bit more about hippuric acid. And so when we think about hippuric acid, I think it's important to kind of know where it comes from. And so you can get hippuric acid from a number of metabolites. And so you can get it from phenylalanine, or you can also get it from a number of catechins. And you get this metabolite through a mixture of both bacterial and host metabolism. And so to really understand kind of what are all the factors that are contributing to variation in its levels, for each metabolite, I'm just showing you for hippuric acid, we basically developed this linear mixed effects models where we carried out these models to really understand the contributions of a number of variables to the levels of these metabolites. And so here, highlighting the contribution of kind of two of the microbial enzymes that we included in the model that are relevant to kind of the production of hippuric acid, as well as just total protein, fiber, fat, and carbs in the study. And what we found is that while the enzyme abundance of this phenylalanine ammonia lyase and then this catechol dehydroxylase did contribute significantly to the levels of the metabolites, some of the other factors that we expected to contribute in terms of host macronutrients played less of a role. And so by doing this for hippuric acid and a number of other metabolites, we started to be able to kind of bin metabolites into ones where diet was enough to decrease the CV versus some of these other factors that we looked at, including macronutrients and the enzyme level abundance of particular genes that we knew were responsible. And so far I've been really just talking about these 4 metabolites, but like I mentioned at the top, we did extensive profiling of a number of bacterial small molecules. And here I'm just showing you the kind of chemical diversity of metabolites that we were able to profile in participants' fecal, plasma, and urine sample. And so this part was really exciting to me because I think one of our bacteria have just an extensive capacity to produce metabolites, and I think it's really important to be able to capture that diversity in order for us to really understand their impact in our biology. And so as you can see here, you can see kind of different levels of benzenoids across the different sample types, or nucleic acids, or phenylpropanoids and polyketides. And so once we were able to do this and do this classification, next we kind of looked at our data more broadly about, well, what do we see, what pops up? And before I kind of show you some of what we found, I think it's just important to have a brief chat about kind of where metabolites go and what that means. And so when we think about plasma metabolites, these are metabolites like they've gone through your GI tract, they've been absorbed, maybe they're modified in the liver, they're in your systemic circulation, and then we're taking a blood sample. For urine metabolites, they've passed through your kidney. And then for your fecal metabolites, these are a mixture of metabolites that may have never left the GI tract, right? And then you have other metabolites that may have gone through the liver, gone through a process called intrahepatic circulation, and then ended up back in the GI tract, right? And so when we think of plasma metabolites, we think of your circulating metabolites. And so what we wanted to do next, the question that we had was like, okay, well, for our representatives of circulating metabolites, how much does the diet contribute to variation in their levels? And so here I'm showing you kind of the first axis of this principal component analysis that we did. And you can see here in the red, this is kind of where all of the participants that completed the study in full stood at the beginning and the baseline period of the study. And so we expected that if there was some significant shift in variation between the 2 time points, you would see that as a shift in terms of position on this PCOA axis. But what we got is, here's the baseline, here's the homogeneous diet, and here's the washout. And so when we looked at the plasma and in the urine metabolites, which again are these metabolites that are systemically circulating, lots of variation. Has nothing to do, though, with our study design. And so that was kind of one of our early clues that maybe there are some other things that we need to think about. And so as we continued on with the study, we also looked at fecal metabolites. And here we saw much more of a greater association between kind of the diet and differences in our fecal metabolites. And one of the other things that we started doing is, and so here I'm highlighting two metabolites, so indole-pyruvic acid and N-acetylputrescine. And for these metabolites, they are representative of a group of metabolites where we found that within an individual, their variation actually increased between the baseline time point and the homogeneous diet time point. And so this was a really interesting opportunity for us to ask, okay, well, if the chemical makeup of what people are eating is the same and we're still seeing this increase in diversity in terms of variation in these metabolites, is there something about the differences in their baseline microbiomes that is predictive of this difference, that's increase that we see? And so since diet was kept the same, we did a number of analyses. And for N-acetylputrescine in particular, we found that the abundance of particular type forms of agmatases, and so agmatases are the bacterial enzyme that's involved in the production of this compound, is that kind of based on the abundance of particular sets of agmatases that you have, that really correlated with how variable this metabolite was in the homogeneous diet phase. And so at this point, we kind of took a step back and looked at all of our data. And so we looked at our microbiome composition data, our microbiome functional data, and our metabolomics data, and asked, okay, well, across all of the study variables that we have, what are the factors that are really contributing to variation and what we're seeing? And what we found is that kind of just general host factors were the most significant. And so if it has a little star, this is based on a Permanova analysis, kind of contributing the most to variation. And so 78% in the species, followed by age, body surface area, biological sex, and the phase of the study. So whether you were in that baseline time point versus the homogeneous diet time point, as well as the actual time point, contributed very little to the overall variance that we saw. And so this led me to the question of, are we looking at diet in the way that we should be looking at diet to really understand differences between individuals over the course of our study? And so kind of stepping back, macronutrients is a really helpful and useful way of looking at diet. But for our purposes, we want to look at diet in terms of the resource space available to gut microbes, right? And that may not mean looking at diet maybe at the level of a meal or looking at diet at the level of macronutrients. And so for me, I really want to think of what is a way for me to capture diet in a way that really thinks of diet in terms of microbiota associated accessible substrates. And for me, that was taking a compound-centric approach. And so for me, that looked like finding a way to basically profiling diet and based on the compounds within it. And so I'll go over this process briefly, but basically what I did is I took our very detailed food logs that we had for these individuals, and so it had meals, it had individual foods, and from a mix of both in-house mass spec data, we have profiling foods in the lab, by combining that data with data from publicly available mass spectrometry databases. So this is things like FoodDB, that has a lot of LCMS and GCMS-based data about small and medium-sized molecules that make up food. I basically took this data to convert participants' dietary food logs into these matrices of compound abundance, and from that you can cluster people into what I refer to as these diet chemical spaces based on their diets. And so in our study, individuals clustered into these 4 different diet chemical spaces. And then from there, you can start to go back and ask questions. And so for example, when we look at how individuals in terms of kind of changes in the abundance of some of these key uremic solutes over the course of our study by a diet chemical space, we see that across the board for hippuric acid still it goes down. But for other metabolites like precressyl sulfate, you have some groups where in diet chemical space 2, between baseline and homogeneous diet, your precressyl levels actually go down, and others it continues to go up. And so we started to see these patterns emerge where based on your diet chemical space, it kind of shapes your responsiveness to other variables of the study that we were really interested in. And so we started to do this with metabolites, but again, I also really wanted to understand, well, how does your kind of diet chemical space relate to your overall kind of microbiota dynamics over the course of this study? And so this is where a lot of network science approaches can be really useful in getting more information about understanding how microbial dynamics over time. And a brief version of how I can explain this is that there are a number of clustering methods that people use already to kind of either associate individuals based on the similarity or dissimilarity of their microbiome's composition. And I basically use a clustering method that's kind of, you can imagine, kind of groups and identifies samples based on how similar the composition of their microbial communities are. And you can basically build this density map, and you can translate this density map into a network where samples move from places of low density to high density on this map. And this kind of tells you what all the microstates are that are available in your microbial community of interest. And so if I were to read this, I would say, okay, well, samples that are represented in this node, some of them travel to this node, and they're traveling this way to these places of greater density. And these individual kind of large clusters or nodes, the bigger ones have more samples represented in them, and the smaller ones have fewer samples represented in them. And so it's a way for me to understand how participants' microbiomes are traveling in terms of microbiome similarity over time. And so this was a really framework for me to start asking a couple of questions that are really important and interesting in terms of studying microbial communities. And so one obvious question that you can ask is just, well, how many different states define a particular microbial community? And so in my toy example here, you can see that you have these different clusters that the microbiomes, that this hypothetical group of people's microbiomes are going through. And then you can also start to ask questions of like, okay, well, does the same person always stay in the same cluster, or does the person move to different clusters? And then do specific taxa always stay in the same cluster, or do they move around? And lastly, do these clusters map onto any clinical phenotypes of interest? And so I went through all of this so that when you see kind of this clustering for our meso participants, hopefully it's easier to understand. And so within this, we identified 5 states, and so this represents how samples in our study are clustering together based on their community composition. And you can see that so there are some samples that are kind of just a few samples, 1 or 2 samples together, that then kind of move into these other spaces where there are more samples that are greater density. You can see that there's kind of this clump of samples together where even though samples are moving from one node to the other, this is relatively connected. And so this one just kind of gave us some information about this kind of first question, well, how many states are there and kind of what are people doing just over the course of the study? And then when we mapped on the diet chemical space to this data, what was really striking is that we did see that participants within the same diet chemical spaces really clustered in the same areas of our map. And so here you can see diet chemical space 1 is really dominates just this one area of this our phase-based network, whereas these other diet chemical spaces, and some of them are joint with other kind of 2 spaces in the same clump, and others are different. And so what was exciting for us to do about this is that we can then go back and ask, okay, well, are there particular kind of chemical substrates within the diet that explains why these particular groups of species cluster together? And that's work that I'm following up on. And so other things that we were able to really look at using this approach is we started to ask, well, how does your diet chemical space relate to other aspects of your microbiome? And so here, what I'd like to highlight is I'm just showing you how much their Shannon diversity relates to your participants' kind of diet chemical space group. And when we did this analysis, one thing that stood out to us is that kind of your consumption of phenylpropanoids and polyketides was kind of predictive of your alpha diversity, which is not super surprising. I think when we think about fiber, I think kind of polyphenols are a component of what you get when you consume a lot of fiber that gets a lot of attention, but I think could get more attention. But it wasn't surprising that you kind of more so than fiber consumption, your consumption of phenylpropanoids and polyketides were a stronger predictor of alpha diversity. Okay. And so one last thing that I want to show you about this kind of component of my work is that because we were starting to using this approach, we were able to just identify more associations between specific subclasses of chemicals that individuals ate and various aspects of their microbiome. I did this Procrustes analysis to basically try to pull out, okay, for an individual sample, kind of what chemical classes within food are kind of associate with either microbiome structure, and so these are the ASV ones, or the genes, or metabolites. And so here I'm showing you some of the kind of significant associations that popped out. And so how you can read this is that, for example, for organic carbonic acids and derivatives in the diet, is significantly associated with your community composition. And when this one circle is kind of a participant's community composition and the other circle is the participant itself. And so using this approach, we started to identify subclasses of compounds where we could say, like, okay, well, these isoflavonoids, for example, also really associate with your community composition, whereas in terms of the particular lipids in the food, kind of strongly associate with your plasma metabolome. And so from this, we started to use this to develop more fine-tuned hypotheses about substrates within diet, that are important for kind of establishing and driving the stability and structure of your microbial community. And so lastly, I just want to share for this component of the work is that when we went back and said, like, okay, well, when we include your kind of baseline diet chemical space into our Promanova analysis of the factors that shape diversity, what we find is that host factors are still, again, dominant. However, we do see that your diet chemical space does significantly shape your microbial community, variation in your microbial community composition, as well as your metabolites. And so I'm really excited about this framework and way of thinking about and quantifying food as a way to start making better associations between the microbiome and diet at a level that's really reflective of these metabolic relationships. And so just to summarize this section of what I've talked about so far is that we've developed this study design that I think was, again, one thing that makes it really useful in the field is just the way and the lengths that we went to really homogenize diet so that in our study participants were really seeing the same chemical makeup. And we used this study to really band individuals and to identify metabolites where, say, diet was significant enough to kind of decrease levels of variability versus factors where, due to our mixed effects modeling analysis and other analyses, we figured out that actually there are microbiome features that really are the top contributors to variance. And so we put metabolites in this bin, but while doing so, we really, again, noticed that host factors that our study was not really designed to measure in detail really do play a significant role in the variability of a good number of these metabolites, and that is something that I want to explore in the future. And then also using this diet chemical space analyses was really helpful for us in starting to answer and address additional questions about how diet can relate to microbiome factors. And lastly, one takeaway from this is that when it comes to understanding variability in metabolite levels, I think it's really important to kind of look at this kind of metabolite by metabolite, or at least metabolite subclass by metabolite, instead of trying to have this general understanding of how diet relates to compound variation. And so next I want to kind of in my remaining time just talk about the approach that I use to really investigate the role of bacterial metabolites in host signaling. And so I have really been focusing on the role of bacterial metabolites in host signaling in the context of chronic kidney disease. And so I think it's important to say a few things about the kidneys. I think they're incredibly an important organ that really shape our quality of life just through their role in filtering out metabolites. And I think it's also really important for us to have just like a shared model of what the kidney does and how metabolites can potentially act as kind of pathogenic stimuli. And so the kidney is made up of these, about 1 million of these functional units called nephrons. And here I'm showing a nephron and the different components of the nephron. And these units are really responsible for filtering, deciding which metabolites kind of get reabsorbed versus which ones are excreted out as urine. And for our purposes, I wanted to just focus on the first part of the nephron, the glomerulus. And so this is the site, which is really the first site at which metabolites enter the kidney. And so just so that you can visualize this, and so the glomerulus includes this, what's called the Bowman's capsule. And in here you have this network of capillaries. And so blood is flowing through with large proteins, small molecules. And then they're flowing through the capillaries. And then these kind of starfish-looking cells called podocytes are responsible. They basically form this sieve that allows small molecules through while keeping large proteins within the capillaries. And so you can see that if you're a podocyte, you're really being exposed to a wide range of metabolites that are coming in through systemic circulation, right? And podocyte loss is actually at the root of a number of diseases of the glomerulus, including scarring or fibrosis. And in this process, podocytes often detach and are lost. And this is really a challenge because you have a set number of podocytes in general. They're kind of post-mitotic cells. Okay. And so I was really interested in understanding then in this context, well, how do these metabolites shape the biology of podocytes in the kidney in general? And we have some really great nephrology collaborators who have done a lot of work profiling, collecting samples that we can profile so that we can use mass spectrometry to profile the plasma, what shows up in the plasma and urine of these patients. And so here I'm just highlighting the kind of top 25 molecules that show up. This was from data that they actually collected, not with us, but with using an outside service called Metabolon. And what you can see is that the ones in yellow are metabolites that are microbial in orange. The ones in blue are ones that are not. And of the top 25 uremic solutes, and so these are the top 25 solutes that are more abundant in individuals with chronic kidney disease versus age-matched controls with healthy kidney function. Most of them are microbial in origin. And they really found, beyond these top 25, there were 91 uremic solutes, some of which are not well annotated, where they had this, that were more abundant in these individuals and with chronic kidney disease. And so I really wanted an approach of, so when kind of thinking of my approach, okay, well, how do I go from this kind of list of 91 plus uremic solutes and many, many others that don't necessarily meet the high criteria that they have for being uremic but are more abundant, to kind of figuring out what are the functional studies that I want to do to kind of shift myself into a more mechanistic understanding of how these metabolites shape human biology. And so using my background, my approach was basically to start off with an in silico approach to kind of prioritize which metabolites I should study and should care about. And so, and I'll describe this briefly, but this is basically of my metabolites kind of asking, well, which ones by virtual screening of how these metabolites may bind to host receptors, which ones are good candidates to think about. Then moved into some other functional assays. And so for me, the main step was going from this in silico prioritization to then doing a number of assays of looking at either GPCR activation in vitro. And then lastly, for the metabolites that I thought were really interesting from this approach, using our lab's really wide, broad collection of human gut microbial strains to investigate kind of what are the species and genes capable of producing or consuming these metabolites, with the understanding that I would then kind of want to be able to modulate their levels in vivo. Okay. And so a few other aspects of this approach that I think are really important to highlight. And so as I said, I had this defined set of 91 uremic solutes. But one thing about mass spectrometry and the targeted mass spectrometry that we do is it's a really great way of measuring small molecules. But one of the things that I noticed is that I would look at, when you kind of look at these metabolites, there are obviously metabolites that were in the same metabolic network that were missing in our data. And they were missing mainly because they weren't measured. And so I wanted a way to kind of systematically identify new potential metabolites that could be important but were just not annotated in our mass spectrometry data. And so in our lab, I do a number of studies kind of in order to understand microbial metabolism, where I basically incubate these large libraries of kind of commensal strands with precursors and kind of look at what metabolites are made and what gets consumed or produced. And so, and as well as in this data set, they had a number of unannotated metabolites where I had basically the mass information about them. I just didn't know what they were. And so what I did is I kind of used a computational approach to kind of predict additional metabolites that were in these metabolic pathways. And I tried to search for their mass in the masses of the data that I've collected. And so in the data that I've collected, I would, for example, look for whether or not a mass was produced when in the supernatant of a sample where the precursor was given, but not in a sample where of just controls. And then kind of look for this mass again in the data here from the patients. And by doing that, I was able to develop a broader network of metabolites that I could then buy and purchase and add on to our mass spectrometry pipeline to quantify later on to actually see if they're there, but also to include in my kind of virtual screening experiments. And then one last filtering steps. And so in addition, so for virtual screening, this involves kind of predicting, looking at G protein coupled receptors and a couple other types of receptors and looking for host binding. And so there are many receptors that you can choose from. And so I filtered my receptors of interest by basically ones that I found were actually present in these podocytes that I'm really interested in and other glomerular cell types based on kind of single cell and single nuclei-seq data from the Kidney Precision Medicine Project. And this was something where Jennifer Harder, who is at Michigan, was really helpful in showing some of their unpublished data about the glomerulus and gene expression. And so I have that as a kind of filter, did some virtual docking, and then moved on to doing a number of assays of agonism. And so these G protein receptors, as you may be familiar with, G protein receptors, they can bind to different G proteins, which relate to different kind of downstream signals. And so for some assays, I was looking at cyclic AMP inhibition or accumulation or calcium signaling. And then I also did some assays that were G protein independent and more looking at kind of beta-arrestin activation. And so I did that to filter things out. And here I'm just showing you kind of some of the hits for different GPCRs that are present on podocytes. And so this is just a tree of some of the GPCRs that are expressed on podocytes, and some of the ones highlighted in orange are ones where we got substantial hits for metabolites, uremic solutes that were in our, that we screened as ligands, and that came up as hits virtually, and that we wanted to move on with. And I'd like to highlight kind of two metabolites, two receptors, because they're receptors that I am interested in the future. And so one of them was the hydroxycarboxylic acid receptor 2, and then another one is this cannabinoid 1 receptor. And for this hydroxycarboxylic acid receptor 2 receptor, this is a receptor that is activated by a number of polyphenol-derived metabolites, including compounds like 3,3-hydroxyphenopropionic acid. And this is a metabolite that I really have moved on to work with to understand its role in the downstream effects of its agonism of the HCAR2 receptor. And so because I was really interested in this metabolite, I then kind of moved on to asking, well, how are which microbial genes and species are responsible for producing and consuming this metabolite? And I've talked about a little bit already. To do this, again, looked kind of for production in the supernatants of our library of microbial strains. And so wanted to highlight there are a few strains that you can think of kind of blue as producing it, and there were kind of a few species that really stood out to me, and so the Eubacterium were among them. And so here I'm showing you data that we had where basically we looked at for which bacteria here, this is data where we can say, okay, well, the bacteria itself has this gene and it produces the metabolite, and I'm just showing you specificity and sensitivity data for this. And so sensitivity just meaning, in this case, the metabolite is produced and they have the enzyme And then specificity in the terms of the fact is like when they don't have the enzyme, they also don't have the metabolite. And so we did this for our data where we have kind of paired genomes for all of our samples. And then we also went into our metagenomic data for a variety of our human studies and asked, well, for how many of these patient samples do we see this metabolite in the plasma? And then we also see an abundance of the enzymes of interest. And so this is work that I'm really excited about because I think it's one important area is really to really expand our understanding of the genes and species responsible for the production of some of these enzymes. And I'm not going to get into this much today, but we have been profiling a number of these phenolic reductases that have been involved in the degradation of polyphenols.
Can I just ask, so did you look at culture supernatants for the panel on the left? It wasn't a correlation with the samples, the fecal samples that had those bacteria, but that was a laboratory experiment?
Yeah, this was a laboratory experiment, yeah.
Thanks.
And then this is kind of human data, so patient plasma samples versus their metagenomics derived, abundance. So thanks. Yeah. Okay. And so just to kind of summarize this section, in kind of looking for and trying to understand the role of metabolites, I really take a structure-guided approach that basically kind of starts with using the abilities of these metabolites to potentially bind to host receptors as kind of the first lens, and then zooming in to exploring their biology. And I think there are a lot of exciting things to do in this area, including, and one of those things I think that is exciting is just how we can use mass spectrometry to kind of expand our understanding of the types of metabolites that are really interesting and important to address. And my ongoing work is really focused on understanding the role of this polyphenol-derived metabolite, 3,3-hydroxyproteinic acid, on this HCAR2 receptor, as well as continuing to profile a number of reductases. I didn't get into this too much that are responsible for their production. And so I kind of want to switch by at the very end of just talking about, well, what do I want to do next? What do I plan to do next? And for me, the big picture question is, again, still, well, how does diet and what are the diet and host mechanisms that really shape microbiome structure and metabolism and interaction with host cellular processes? And there are kind of two big questions that I'm interested in asking that kind of represent the two big research directions of what my future lab would be. And so one is really zooming in and trying to understand of, well, how do these podocytes that I talked about very briefly sense and respond to these metabolites? There are a number of things that I to do in that regard and that level, and also to really dig deeper into how do diet, host genetics, and microbiota collectively contribute to MBM levels of production. And so just to say a little bit more about this, thinking about how do I get at this podocyte question, one part of that the question actually has less to do about the podocytes and more to do about the microbial chemistry. And so one of my big interests is really to continue my work in understanding how these metabolites that I think are biologically interesting are produced. And so for me, that, again, is kind of continuing my pipeline of culturing bacteria and anaerobically and understanding and figuring out those species and genes. But also, I think a really important next step that I'm excited to spend more time doing it on is just understanding how we can modulate their levels. And so the ways that I envision doing that is, one way is, again, through diet or through by enzyme inhibitors targeting specific enzymes. But also, one of the things that I've been slowly getting involved in the lab and really excited about is just through defined communities. And so what's really nice about having a defined microbial community in comparison to, say, colonizing a germ-free mouse with like a fecal sample is that you know what you're starting out with. And I think the really exciting area is just our ability to kind of add and subtract members from a community and understand how that affects the production of an enzyme or a metabolite, of interest. Another area that really relates to this, again, is just kind of zooming in on their effects on podocyte biology. And so I didn't get through this much today, but HCAR2 plays important roles in really regulating kind of inflammatory pathways in podocytes that are relevant in the context of chronic kidney disease, just in terms of how much and whether or not these podocytes secrete some pro-inflammatory cytokines that can then recruit a number of immune cells, mostly macrophages, to the kidneys. And I think that's an important area to really study. And because the receptors that I'm interested in are not just expressed on podocytes, and so your HCAR2s are expressed on endothelial cells and on a number of another different cell types, I plan to move this work and into podocyte, using mouse models with podocyte-specific knockouts of my two receptors of interest, and I'm happy to talk about that more if people want to know more about. And then on the other side, another research direction that I'm really excited about is that, again, just understanding and getting better at predicting how people respond to diet. And so this whole idea of precision medicine has had a number of hurdles in terms of our ability to really predict how, when you give someone something to eat, how will they respond. And I think a number of people have made really great contributions to tackling this. But I think where I want to contribute is, one, just in terms of expanding the types of metabolites that we can quantify, and then two, I think one thing that I hope that I've demonstrated today is that when it comes to these microbiota-dependent metabolites, is that you do have to kind of consider the host and what the host is doing. And so you can consider the host in terms of the host kind of regulating the microbial community through immune responses in that regard. But I'm also interested in just understanding the contribution of host metabolism. And so my interest is really in focusing on how these drugs, how these enzymes that modify and conjugate compounds that are mainly in the liver and transport these molecules also play a role in their abundance. And so that's something that I will focus on a bit more in my lab. And just to kind of give a long-range vision, I know that we're kind of low in time. I think I'll skip here. Is that what I think is really exciting just about types of questions that people may want to answer, and like things that you may want to train in, is that I see there are so many kind of routes for people to take. And so I think one of the key challenges that the type of research that I wanted to address is just thinking about microbiome-centric methods for really understanding nutrition. And this is a really fun mix of using bioinformatics and chemoinformatics to get at how we can quantify diet in these ways that integrate mass spec data with other data types. Two, I think decoding microbial chemistry. And so this is always going to be kind of a bottleneck. We need to be able to manipulate these, to understand and also manipulate microbial chemistry in vivo. And so I think molecular genetics is always going to be a really important tool. Cell signaling, I think there's many questions related to cell signaling to understand. And so I'm thinking about cell signaling in the context of podocytes, but not only is there a lot to think about in terms of activation. I think many of these metabolites are being ignored, and understanding those processes is important. And so I think cell signaling is another exciting area. And then just kind of getting to variation mechanism. And I think that these types of studies are really setting me up and others up to really understand, well, what is the mechanistic basis by which kind of these metabolites can contribute to both outcomes but variability that we see in disease. And of course, at least for me, the end goal is always to kind of make this mechanistic work kind of translational and thinking about how we can use this in a preventative manner. And so with that, I'll just wrap up with acknowledgments. And so I'm currently a member of the Sonnenberg Lab, and there are a lot of great people to collaborate with. And so all the people in Bold, Sean Spencer, Sproul Hand, Will Venturin, Brian Yu, Brian DeFelice, and Juan, these are all kind of other trainees that were really helpful in some aspect of something that I've talked about today. Tim Meyer is the main nephrologist that we collaborate with, who is really helpful, and Natalie Plummer, who's a part of his lab. Dahlia Perlman is a really great dietitian who manages a lot of our dietary studies. And then, of course, the feedback from Michael Fishbach is also always helpful. And lastly, just acknowledging Justin, my mentor, who's really great, and then my source of funding, which is the Hannah Gray. And with that, I'll take any questions.
I might have missed this, but did you mention the extent to which the microbiome changed as a result of your defined diet?
Yeah, and so I guess it depends on what aspect of the microbiome you're thinking about.
By like bacterial and fungal diversity.
Yeah, and so if we look here kind of at the Permanova analysis, so I guess I should say two things, is that we did see changes in the microbiome. They were not related specifically to the intervention, and so the intervention was not a driver.
Do you think if the intervention was longer, there may have been like a convergence on a microbiome?
I do not think that that would have had a difference. I think that if we had done an intervention that was, and so our intervention was designed to kind of recapitulate the macronutrients that an average kind of adult living in the U.S. eats, just based on national nutrition data. And I think what we've learned so one part of it is like it's a short-term diet, and short-term diets have short-term changes in the microbiome. But I think that, and what we found is just unless we did something to really target or eliminate specific kind of micronutrients, I don't think we would have seen the change. Yeah.
I was just wondering, based on your DCS findings, if like you have a homogeneous DCS group, like you're all getting fed group 1 or so, if they all begin to converge their microbiomes, like group 1 microbiome or something.
Yeah, what I think what we learned from the DCS is that your kind of habitual dietary intake shapes so much of how you respond, and so like unless we, I guess we would have had to really kind of change your diet, everyone's diet in the same ways for a longer period of time, and then done our intervention for them to be the same. But yeah, I think there's so much variability that at least a short-term diet is probably not going to make a difference. Yeah.
Actually, glad you have this slide up. I had a question about this one too. So I was looking at the baseline DCS and the explanatory value of species versus genes. I was just wondering if you could talk a little bit about why sort of gene composition seems to be doing a less good job. Do you think it's more related to kind of core functions of specific bacterial species?
Yeah, I think that, so at the gene level, you'll notice that there's just really there's not a significant contribution of the baseline diet chemical space versus the community composition level. And I think that's a number of things. I think one is just, so some of it is technical in the sense is that you're doing these and there are so many genes that you're measuring, and there's, and so you have a low signal there. And then another thing, I think there's just this functional redundancy. And so I think that like, if say I zoomed in on particular classes of genes, maybe I just focused in on carbohydrate active enzymes or something like that, maybe I would have seen a clearer signal if I could have kind of broken it up by KEGG pathways or something. But at this broad level where so much kind of functional classes are included, I'm not surprised that we saw so little of an effect. Thanks for asking.
Do you have any strategies to get at chemical signatures for HCAR2 activity in urine samples so that you could, in one fell swoop, see if the metabolites are affecting its function?
Oh, so that's interesting. And so do I have chemical strategies for getting at kind of receptor-mediated activity that I could kind of non-invasively do? And so, I guess there are a couple of ways to think about that. And so one thing that I didn't mention is that kind of when you have chronic kidney disease, these kind of podocytes detach and they accumulate in your urine. And so that's kind of proteinuria is one thing that happens where you know that you have podocyte loss because you see them in the urine. And I think the output of HCAR2 signaling is mainly, and so there are kind of two hypotheses that I have. And so one of them just relates to a transition from kind of, I guess, basically I guess there are changes in its ability to, through the NF-kappa beta pathway, kind of change to regulate pro-inflammatory versus anti-inflammatory signaling. And so I think systemically I could kind of just, or regionally I can kind of look at differences in pro-inflammatory cytokines and kind of use that as a marker of HCAR2 signaling. But in the urine, I guess one thing that we do is kind of you look for podocytes in the urine and see how they're doing. Yeah. Is there another question?
out of time and I have you for longer of a day, but I'm also just curious how abundant either a species, with enzymes has to be for the molecules to be detectable in the plasma, right? So if you have I'm assuming phenylalanine or something like that precursor is abundant enough that you can see its derivatives, but for a specific microbial activity, there's all these interesting kinetics, from the ability to make it to the plasma in addition to its creation. So, do you think that there is a limit with your current methods or a back-of-the-envelope calculation that determines what types of molecules you'll see or what pathways are sealed?
Yeah, and that's a really interesting question because you have some metabolites where the species that produces it, like you'll have, say, C. sporanoides, that is like the one bug that produces some very weird version of a bile acid, and it's 1% of your microbiome, and it makes a lot of it. And then you have the kinds of compounds that I'm interested in where they're polyphenols and they're often conjugated to a bunch of sugars, and so they have to actually reach your colon, and that takes some work and deconjugation by other bacterial enzymes to kind of increase bioavailability. So there are a number of steps that kind of shapes how much actually enters kind of the colon where we think most of this is happening, and that varies a bit. But we do see it just from healthy participant samples. We see a lot of these polyphenols at the low micromolar level in their plasma, so we know they're getting there. I think the trick there is that we have to distinguish what is just diet versus microbial made, and so some of these metabolites, they can come from microbial processing and they also come from the diet, and so we have to do a better job of distinguishing that route. But that is, it's always something we have to consider.
Yeah. Great. Well, it's time. So you can join me in thanking Leah one more time for